nucleotides, we got a phosphate and a deoxyribose. That's the sugar. There's adenine, thymine, cytosine, and guanine rolling up on the scene. And there's your cell in mRNA. You got that? Okay.
Helicase is the unwinder, topoisomerase, twist finder, primase initiates at the origin, DNA polymerase elongates twin, leading, lagging, continuous, sagging, Okazaki. Fragments connected by ligase, nucleotides take their place. Telomeres are the stoppers. Don't confuse with hip hoppers. This ends the semi-conservative process by which we live.
Transcription begins with initiation, then it continues with elongation. Turn it around in termination. What? Now, initiation activation is step 1. Attach to a promoted region, get some. DNA zips into 2 separate strands. Now we're running transcription firsthand. Elongation as the polymerase unzips DNA and assembles RNA. One strand of DNA is a template. 5' to 3' is no mistake. RNA nucleotides are only one produced, as one strand of DNA is 2. This is what differs from replication. Now let's move on to termination. Termination marks when 7 A's halt the process of RNA. This special sequence of nucleotides puts transcription on the backside.
But for the messenger, at least a molecule of 5' cap becomes useful tool. It's a guanine nucleotide with 2 extra phosphates, stable and wide.
3' in is the poly A tail. 200 adenines make it up well. The tail guides the messenger out through the nuclear envelope route.
Then the splices start kicking in, cutting the exons in, introns are open, exons express, introns are hidden, small nuclear ribonuclear protein delete, introns making them forbidden.
The messenger has one destination, to the cytoplasm with no hesitation. Our RNA binds it to the ribosome, so it no longer has to be alone.
After transcription comes translation. There are 2 steps: initiation, elongation, and termination. Initiation, tRNA brings anti-codon, matches with codon. Elongation, amino acids brought the mutation. Translocation, from the A site to the P site. Repeat until termination. A stop codon is the negation of this process. Oh yes, all this is the polypeptide, so go high. Proteins are a wide variety in our society. First we have channel, passages annual. Next is carrier, specific molecules acting as a barrier. Also aquaporons that lets water in. Peripheral, on the outer surface. Integral, extended into the space. Transmembrane is both. Fluid and mosaic explains this oath. First degree is linear. Second, hydrogen bonds. Third, disulfide bonds. 4 is 2 chains together. Example is steroids, which you need to avoid. This is the nerd herd. Word.
DNA to protein. The stairs represent the helicase unzipping DNA. Volleyball match represents the complementary base pairs, which are on the same team. The fighting scene represents initiation, mRNA, like it is initiating war, just as initiation, transcription. Terminating thymine is just as step 3 in transcription. Your cell replaces thymine in the volleyball match. Adenines match. The car represents translation and drops off anticodons. And dancing is basically making proteins. Yay!